right. Everyone get one? You needed one? I need it. I printed out on both sides. That saves paper a little bit. Don't know if we needed to or not. There's a bunch of pages, but I do have a couple extras if you want to grab it. Do you want one?
Yeah, I can grab one.
And then it's up to you. I didn't know how you would store it. You can 3-hole punch it if you want. You can leave it as is. It's up to you. I don't know how you're going to organize it. All right, don't forget we have a quiz over the terminology, the intro terminology, host names, parasite names, and so forth. So review that. That's due tonight by 11:30. Get it done. All right. This is where we left off. We're introducing platyhelminths, the flatworms. All right, they're dorsal ventrally flattened, bilaterally symmetric, acoelomate organisms. Their digestive system consists of an incomplete gut, or they can have no gut. It just kind of depends on which group we're in. All right, so the flatworms, the digenetic trematodes that we looked at in lab, they actually have an incomplete gut. So you've got a gut, but it just ends. All right, it's a blind gut is what we call it. And that's different than what we have, right? We've got a gut, or we've got a mouth, and then the gut goes through and ends at the anus. We'll see that in the nematodes. Has some implications. What's an implication of this, of having a blind gut? What's an implication of that?
It goes in and out in the same place.
Yeah, so whatever you take in has to go out same place, right? You have to basically vomit the waste. What else?
Limited food options.
What do you mean?
Probably can't eat a whole lot because you can't hold a whole lot.
Okay, so you're limited to, yeah, you can't just keep stuffing yourself. I mean, you've got to at least regurgitate it at some point, right? The other part of this is, since we have a blind gut, we really lose the ability to have, I don't want to say compartmentalized digestion, but digestion that happens along different points. So like for us, we've got stomach, that has a function. Small intestine has a function. Large intestine has a function. We've kind of divided up what's going on in different parts, and we don't necessarily have that here. So there's kind of a benefit of having an anus in terms of digestion. Do you need one of these? I printed out the handouts, the note outlines.
I'll take one if there's one.
Oh yeah, I printed out 15. We're good. All right, so that's digestion.
Thank you.
Some of our worms have no gut. So example are the tapeworms. What's the consequence of that? We don't have a gut. What do we have to do? If we have no gut, we have no mouth. What does that mean for our organism? Okay. Diffusion or
absorbable nutrients around you.
good generalization. Yes, absorbed. All right, so yeah, some of it will happen through diffusion, some of it will happen through pinocytosis, some endocytosis reactions. Yeah, if you're in there, you have to absorb your nutrients. That's how you get it. What kind of adaptations do you think we're going to see in those organisms?
High surface area to volume ratio.
All right, so we're going to have some adaptations that increase the surface area. The more surface area, the more area you have that you can absorb. All right, so moving on is the excretory system. Excretory system is primarily osmoregulatory. All right, that's its primary function. All right, its primary function. Our excretory system, the osmoregulatory system, is what's called a protonephridial or a flame cell system. And it's named for these flame cells, flame bulbs. All right. So protonephridium is like an early stage of, you could say, a kidney cell, if you want, functioning kidney. It functions in much the same way. What it's going to do is take in water through some slits in it. All right. And then as the water traverses, it will reabsorb nutrients, and the waste proceeds down through a collecting duct to an excretory bladder or directly to the excretory pore. All right. So in this, it's called this flame cell because the way the water moves in is through these flagella that beat kind of in a synchronized pattern. When we look at these things underneath the microscope, it looks like it is like a candle flame, hence its name, a flame bulb or a flame cell. The beating of the cilia draws in this water along with ammonia waste, and then we reabsorb it down in more of our collecting ducts or collecting tubules. All right. Its purpose really is osmoregulatory. It's going to filter out ammonia waste and kind of regulate what water you have in there. So with some of the worms, I hope some of our snails that we pick up this year have cercaria, and then I can get some of these cercaria onto a slide and kind of get them in position long enough to where we can look at some of these flame cells. But what we see is that if we're in a more of a freshwater environment, you will see these cilia or these flagella beating much, much faster than if we're in more of a closer isotonic solution. Because these things, water's rushing into them. They'll explode if they don't get rid of that extra water. All right, so hopefully we can see some of these. Any questions? Nervous system. This applies to trematodes and cestodes. All right. But this is a ladder type system. So we've got ganglia. We've got a cerebral ganglia. Usually two, can't have one. All right, then from those central ganglia, we have longitudinal nerve cords that run down the system. They can be connected at the end, or it could form this. So it could be connected, or it can be not connected. And we see these lateral commissures, transverse commissures. So lateral branches that connect the two sides of those nerve trunks. All right, we refer to this like ladder type system as being orthogon, an orthogon nervous system. Now, coming off of these aren't just for movement. They also have sensory organs tied in with the nervous system. This is kind of how they sense their environment. All right, and the sensory elements that we have include tactile cells, chemoreceptors, eye spots, so light detection, and statocysts, which is all about orientation. So these are all connected. Where they occur varies based on our species. So the tactile cells are those to detect basically surfaces. It's our mechanoreception. They could be part of maybe appendages, maybe protrusions on the worm. The chemoreceptors are normally pits on the surface. We're detecting chemicals, and yeah, some of these worms, they utilize chemicals, chemical trails to find their host, or in some cases, while inside the host, to find the organ that they need to get to in order to mature. Some of our worms will have eye spots. These are light detection. These are light detection. Some of our slides that have the cercaria, yeah, they've got eye spots on them. It's all about light. And then others have these statocysts, which are they're for orientation, but they work by gravity, is what we're looking at. So the worm kind of has an idea of where it is and how it's positioned relative to the surface of the earth. misspelled that. Should fix that. All right. All right, so the reproductive system. There's a basic pattern in all of our platyhelminthes. So this includes our trematodes, our monogenes, Aspidobothreans, and our tapeworms. All right, so what we're going to do is kind of introduce this reproductive system, the terms and the structure, kind of set up the arrangement, knowing that sometimes the arrangement varies a little bit based on what group we're working in or what family, what species, and so forth. All right, so in our platyhelminths, most of our species are monoecious. What does that mean?
Just a single reproductive organ. Wrong? Or they
They have both. Single individual that has both sets of reproductive, both male and female. I think you probably had. You misspoke. We'll go with that. Right? So most species are monoecious. So we're going to see individuals that have ovaries and testes. Right. Now, even though they're monoecious and they can self-fertilize, usually cross-fertilization is the rule. All right, usually that has to happen in order to reproduce. If they're in a bind, they could self-fertilize, usually. But normally, we're dealing with cross-fertilization. And there's different ways that they kind of accomplish that, kind of control how they can cross-fertilize. We'll talk about that a little bit with our tapeworms. All right. We do have some dioecious species, though. So those are two, species where you have separate sexes for the worms. And in that case, yeah, you have to cross-fertilize. I mean, you need a male and you need a female. They need to get together in order to produce the eggs. When they do mate, sperm transfer happens where the serous projects and deposits the sperm into the female reproductive tract. All right, this is typical cases. We had dichrocoelium in the lab where some of those specimens have the serous actually protruding. You're going to see some tapeworms. We have some tapeworm specimens where the serous is protruding. That's going to enter the female genital system, the female reproductive system, and the sperm will be delivered. The sperm will then travel, oftentimes through the uterus, at least for the diogenes, to get to the ootype where fertilization can occur. Along the way, it could be stored in a seminal receptacle, but not every worm has a seminal receptacle, or not every species has a seminal receptacle. Some of our cestodes, however, practice what is called hypodermic impregnation. All right, so now our serous isn't entering into the female reproductive system. Instead, it's piercing the tegument of that worm. And then the sperm gets delivered right into the parenchyma, that spongy connective tissue. The sperm finds their way then to the ootype. All right, once we have fertilization then, you're going to have egg development. And in the parasitic species, these eggs are ectoleucithal, which basically means that the nutrients for the egg yolk are all provided by other cells, by cells other than the ovum. So if you had, I don't know, did you talk about some of the development in like introductory biology? I'm sure development. We had development last year, right? Was development taught last year? Nobody took it? So oftentimes you have your the egg cell itself, the ova, will provide the nutrients. You'll have development of the nutrients in those cells. In this case, we have separate cells providing the yolks. So that's ectoleucithal. All right, and that's kind of, I present this term, I'm going to rarely ever ask it, but this ties into the vitelliaria and the vitelline glands. They're providing the nutrients for the egg. And then part of the reproductive system, we can also reproduce asexually. And this is different than our self-fertilization. With the asexual reproduction, what we're talking about is worms just basically cloning themselves. You have germ cells that just ultimately mass produce the next larval stage. This is common in trematodes. Some cestodes also do this, and we'll talk about why they might do that. Ready? All right. So traditional. We'll get to the terminology of the reproductive systems, the different parts, in the when we get to the. I don't know when we get to it. I thought I had it. Fast forward. Yeah. No, I guess not now. We will. We'll get to it. We'll see. Maybe in the Trematoda. Intro to Trematoda is when I do that. All right, so systematics, traditionally there's been 4 classes. Traditionally. Break it down into Turbellaria, your Trematoda, Monogenoidea, and the Cestoidea. Cestoidea, or cestodes. Traditionally. We've since kind of broken it down and said we've got the Turbellaria as their own group. These are the free-living flatworms. And then the parasitic species form a group called the Neodermata. Right? Neodermata is called that because of the new skin. So it's part of the transition to parasitism that we have to find some way where we alter our tegument to maximize absorption, because in some cases we are losing our digestive tract. All right? So all of our parasite species all fall within this Neodermata group. It's like a sub-superclass. Members of this have ectocythal eggs. We have a loss of a ciliated epidermis, and that's kind of a trigger. So in the turbellarians, you have a ciliated epidermis. You lose that in our parasitic species. But instead of that ciliated epidermis, what we now have is what's called a syncytial tegument. All right, and we're going to describe what that syncytial tegument is. And that is our new skin, the Neodermata. So as part of this Neodermata, we're going to look at the class Trematoda. In this class, we have suckers. These are going to be primarily adhesive organs. We have a male genital pore that opens up into an atrium, and adults have a pharynx near the oral sucker. So we've got a mouth in this group. Now, in this class, we have 2 subclasses. We have an Aspidobothrians and the Digenea. We looked at Digeneans in the lab. We're going to talk about Aspidobothrians, and we've got some slide box appeared that has an Aspidobothrian in it. I don't know how good that specimen is. A lot of those slides have clouded up, so I'm going to have to see what I could do about getting them remounted. There's a lot of cool trematode slides in there. All right, but we're going to talk about these. And a key difference between the Aspidobothrians and the Digenea is Aspidobothrians have this large modified attachment organs, and we're going to see those. And I'm not going to ask you, well, what's the difference between Digene and Aspidobothrians? We're not going to ask that. Second class that we'll talk about, the second parasitic class are the monogenes, the Monogenoidea. Difference in the Monogenoidea is larval stages. So there's difference in the larval stages among all of these groups. There's different names in the larvals with the larval stages. The adults always have a single testis in this group. And all of them, well, I should say all are ectoparasites. The endoparasites are derived. There are a few that are internal, but almost all of them are ectoparasites. And then the cestodes are our tapeworms. I mention it here because the classification has been changing. It's getting away from like the class, order, family level, and instead is starting to use cohorts instead of class. And it could be due to the, you can say, messiness of the trees, of phylogenies. Things that we thought were, yeah, these are all related. Once you start doing sequencing, they're not. They're in multiple clades. So we're going to use cohorts because that's what some of the newer textbooks are doing. And we got 2 major cohorts, the Gyrocotylidia and the Cestoidea. I believe the Gyrocotylidia was one of the options that we could talk about. Key thing is that our regular Cystoidea are the tapeworms. It has those the strobilization that occurs. It has those presumably segments, the proglottids. The Gyrocotylidia don't. It's basically one individual. It doesn't have the segmentation. And they really are pretty cool. You look at them, you think, wow, that's a parasite. And then you're like, wow, that's actually a tapeworm? Yeah, it's part of the cestodes. Part of the cestodes. So that's what this means. Adults are polyzoic or polyzoids. They're going to have all these segments on them. All right. Important part is this new skin, the Neodermata. So all of our parasitic groups are monophyletic. Right, they're all monophyletic. And the big change was this transition from having a ciliated epidermis to having the syncytial tegument. Right, so with our ciliated epidermis, the epithelium is thin. It has a single layer of cell. All right, anything that comes in has to go through those cells. Now, the cell, that layer of cell, will include glandular cells. And you also have duct, so that these worms can secrete mucoids and other substances out to the environment. In the Trematoda and the Cystoidea, we lack that ciliated epidermis. Exception, though, is that sometimes we have a ciliated epidermis in our larval stages. That's kind of where the turbularian relationship comes into play. But in our adult stages, we have the syncytial tegument. So we don't have a single layer forming our epidermis anymore. Instead, all of the cells are together, forming one continuous, essentially cytoplasm, you could say. All right. And the individual cell bodies, the nuclei, are located in pits. So the definition of our syncytial tegument is a multinucleated epidermis where the nuclei are located in cytons below the superficial muscle layers. And I have a diagram to go over. And it has important roles. You ready? About? I don't know if I have it different. Ready? All right, so here's the structure of our syncytial tegument. We've got, this is exterior of the worm. We can say that we are out, this is the lumen of the gut. All right, this part is our tegument. We have our cytons. The cytons is where we find our nuclei, and we find our mitochondria, our ER, and all that stuff. Things that you would typically find in the cell. They are isolated in cytons, and they are located below our muscle layer. All right? These are not separate from the epithelium. We actually have these canals, these spaces that connect the cytons to the exterior part, to the distal part of the body. All right, that channel is called an internuntial process. So then once we get to this distal layer, it's anucleated. There are no nuclei in this zone. So we have our production, synthesis of compounds, secretory compounds, processing. You have vesicles that transport it up to the surface so that they can release it. All right. At the same time, we have endocytosis that can occur, penocytosis that can occur, that'll bring vesicles down to the cell, to the cyton itself, or can be transported right to the parenchyma where the contents get dumped and then get transported, diffused through the rest of the body. Along with this tegument, we have a couple different things that could appear. One, we can have spines. We have a worm where we can zoom in on the edge to see these spines. They protrude through the tegument. Normally attached with muscles, so some of these can actually move the spines. Why might they be useful? Depends on on what stage they're in as to what their primary role is. If we look at adults in the GI tract, it's thought that these spines may actually act to help hold the worm in place. So the worm kind of gets buried in with some of the epithelial membrane through your villi, microvilli, and the spines, because they're pushing back. If you arrange that worm, it's going to have a hard time to get pushed back towards posterior part of the gut. If they're on, let's say, a larval stage, a cercarial stage that is penetrating in, if those spines are pointing back, all that the worm has to do is kind of get that part in, and then it's going to be very hard for the host to get it out. It's just going to keep moving through into its area. Also, with part of this tegument, we can have sensory structures. This is a papilla, but it's sensory in nature. All right, so what is the role of this tegument? We've got three main functions. One is digestion. Our cytons can produce proteolytic enzymes that get released, and then those enzymes break down particles outside the host into compounds that could then be internalized. All right? Some of our worms, they don't have a mouth. Our cestodes, they don't have a mouth. Everything that they acquire has to occur through absorption. All right, so digestion, important role of our tegument. Absorption is number two. So it will absorb some of these nutrients. It will absorb water. It'll, stuff that it doesn't have to break down. It can absorb it, and it can absorb it through either endocytosis, penocytosis, or diffusion. So what's penocytosis? I've used that term. What is that? Yeah, you say cellular drinking. Yeah, it's a small scale endocytosis is probably maybe a better descriptor. But, yep. All right. And the third thing, and this could be the most important part, it's protection. So these are parasites. They're inside. They are getting hit and bombarded by the host immune system. And we can ask, hey, why is this an anucleate layer? Well, this layer is anucleate because it's going to be constantly sloughing off. All right. On the surface, we have a glycocalyx, which is basically complex carbohydrates, proteins, and stuff forming a protective layer, and it's constantly replenished. So as host antibodies and host cells are coming in and binding to the surface, they just fall right off because that glycocalyx gets shed, but then gets replaced. And this glycocalyx extends across the entire surface of the worm. So even on the spines, you have that thing. All right. So the sloughing of the glycocalyx plays a pretty important role. Antibodies bind to it, but then those antibodies get whisked away. It's still bound to their targets. All right. They're still bound to the targets, but they're no longer attached to the worm. Even in the gut, our antibodies are hitting those worms. But the antibodies just get washed away because glycocalyx is left off. The other part of the protection is that they can secrete polyphenols and mucopolysaccharides. These things can inhibit host enzymes. So if the host is dumping enzymes to try to break down the surface, to break down the tegument of this worm could have a defense mechanism that tries to bind to those and try to inhibit their action. All right. So if we didn't have this, if we had just a single cell, epithelial cell, you can't really slough off the cell membrane. So if an antibody binds, it's going to bind there. You're going to have the effector cells coming in to try to damage the parasite. But in this case, you've got a glycocalyx that forms that outer layer and it's just constantly replenished. It's sloughing off and taking everything with it. Question, it's an important part of this group. So make sure you review it. All right. So that wraps the introduction up. Log into Blackboard. There it is. is blame the state. I was going to blame the university. Why do that? No, it's a state law. State law requires this now. Two-factor authentication. Basically do anything. So you gotta be kidding me. All right, so quiz 2, platyhelminths introduction. We have one that's due tonight.
Do you have any slides up on Blackboard?
Mm-mm.
The one from here?
No, just the immunology one. I do have all the figures and stuff are on a separate handout. So the figures are posted. And you can copy and paste on here if you wanted. All right, so I think what we'll do is make this alive. We'll set this due date Friday, 11:30. Get this done. Actually, we'll do Sunday night, the 30th, 11:30 at night. Gives you time. And it's live. All right. Next up. So that was the introduction. What we're going to do is hit it down to our classes, individual groups, and we're going to start with the Aspidobothreans. All right, so we're in the class Trematoda, so still phylum Platyhelminthes. Class Trematoda, subclass Aspidobothrea. And this should be in the packet. This part should be in the packet. All right, so the Aspidobothreans are a small group of parasites. There's only about 80 known species. About 80 known species. We present it because of their interesting life cycle pattern. It's kind of a question of, are these parasites that are adding a host, or do we have some of these groups that are losing the host to become, let's say, a one-host life cycle or even a possible free living cycle? So we've got some interesting life cycle patterns here. These are all endoparasites of mollusks, fish, and turtles, or turtles. Right? So don't think that once we get into mollusks, we can also infect fishes and turtles. It's just this group, this is who they infect. It's actually a poorly studied group. It's not necessarily poorly studied because we only have 80 species, but because there's very limited medical and economic importance to them. If it doesn't infect humans, why study them? That's kind of the big part of it. These have no impact on our food supply. They have no impact on our health. So there's no NSF or NIH money out there to study them. Now, members of this group have 3 distinct body forms. All of them have a large ventral sucker. All right? And that ventral sucker is going to be modified in these 3 body forms. So this large ventral sucker, this huge, or a ventral sucker, just another name of it would be an opistaptor. And we're gonna see that term appear. It's kind of more of a generic name. But in this group, that large ventral sucker is called a Bayer's disc. Bayer's disc. All right, so what are these 3 distinct body forms? Ready? All right, so we have body form number 1. We have a worm that has a single large Bayer's disc that's divided into shallow depressions by muscular septa. So these are diagrams. Here's our single large Bayer's disc, and that's our Bayer's disc. This is our large ventral sucker. It's modified into these depressions that are connected by septa. And you've got variation in the arrangement pattern. All right, so the depressions can be called alveolus or alveoli, or we can call them loculus or loculi. Single versus plural. I probably prefer loculus and loculi, just because we refer to alveolus and alveoli to structures in our lungs, or at least it's what I will be using, right? We do have some worms that go through our lungs. We'll talk about those. All right. This kind of acts as, gets onto the host, and then the muscles contract. And when they contract, they form the suction that holds these worms in place. So, Aspidobothrea is, I think, one that I might have a slide of. So we're going to check to see if actually do that, and then it would be actually pretty cool if we did. So that's body form number 1. Body form number 2 has that Bayer's disc modified into longitudinally arranged suckers. So we're not dealing with an oral sucker and a ventral sucker. Instead, we're dealing with numerous ventral and posterior suckers. So in this example here, Syncytial, right, we've got these suckers that are basically arranged all along the length of that worm. So that's body form number 2. So body form number 1, that's our Bayer's disc, has the loculi with the muscular septa in it. We have longitudinally arranged suckers. That's our body form number 2. And then body form number 3 has our Bayer's disc kind of lacking in a way. So there's no obvious sucker or attachment. Instead, it's been modified into more of like an accordion-like structure. It's basically consists of transverse ridges called rugae. All right. So there's no septa, no suckers, no loculi or anything. And these rugae then, when they contract, can kind of like grab and pinch. All right. So you've got suckers, I think suction cup and everything. Loculi kind of do that same thing. But on these, we're just kind of, when we flex, we kind of pinch and can hold on that way with the pinching. Because you've got numerous pinchers along this way. So that's kind of like an accordion-like structure there. Right. 3 main body forms. Ready? All right, life cycle in this group, they all have a direct life cycle. Well, most have a direct life cycle. Some of them utilize an intermediate host. Sometimes that intermediate host might be optional, or might actually be kind of like almost like another definitive host. I think that might be one of our examples, our example life cycle. All right, so notably, those worms that are parasitic in a vertebrate host, they are going to use an invertebrate, or they do require an intermediate host. If we're in a mollusk, if that's our definitive host, we might not require an intermediate host. So the generalized life cycle pattern for this group is that we have the egg. The egg hatches, releasing the larval stage called the cotylocydium. That cotylocydium then develops into an adult. There's a generalized pattern. If you recognize this is the parasite as an Aspidobothrian, and I ask for a life cycle, you can get partial credit just by having the correct order. Egg, cotylocydium, adult. Okay, we're back to egg, cotylocydium, adult. All right, so what is that cotylocydium? This is our larval stage of the Aspidobothrea. All right, its features is that it has ciliary tufts that are used for swimming. So we've got these tufts of cilia located on the body, and that's different. Some of our larval stages, this will be covered in cilia, like a complete ciliated stage that it uses for swimming. Not these guys. The cilia, instead of being covering the entire organism, they're arranged in these tufts. You can think ponytails on these guys. All right. These larval stages also have a mouth, a pharynx, a simple gut, and a prominent posterior ventral disc. All right, we don't have the loculi in it. All right? Some sort of disc. All right, and again, this is different than the other platyhelminth larval stage, and this is the first larval stage after the egg. And we typically don't see a mouth and pharynx and gut in those larval stages. Normally they're just germ cells, that's it. But these guys have a bit more development. And then we have this single disc. It's from this one item here that gives rise to the attachment organs of the 3 different body types. All right, so if we're going to have suckers all along the length, so body type number 2, this will just keep dividing into additional suckers as this worm matures and elongates. Other times this will start to differentiate into that actual Bayer's disc. Like we said, it's that anterior end of the disc. So I think all of ours, anterior end at the top, posterior is at the bottom. So when it swims, it's going that way, going towards our ceiling. And lastly, there are no hooks in this stage. We'll see hooks in some of our other larval stages. Kind of a distinguishing feature. And in this stage, there's no asexual reproduction whatsoever. Not in the life cycle of these parasites. It's egg, cotylocydium, adult. All right. So what we're going to do is introduce Lobatostoma manteri. Lobatostoma manteri is our example life cycle of the Aspidobothreans. Here. All right. Gonna leave those up. What we are going to do is you're going to get your first life cycle. We'll see. We might be able to get through it in 5 minutes. Maybe. All right. So, mute it. Probably what, anatomy? Maybe anatomy? Is anyone in anatomy this year?
No, but I remember when I was in that meeting, I thought was exactly.
Cool. You remembered it. It wasn't like nightmares. All right, so we're dealing with example Lobatostoma manteri. We're going to present this because the life cycle is rather interesting. Right? Specifically, it has an extremely host-specific life cycle. Right? These are going to infect fishes down on the reefs outside of Australia. The adult worm is in the snub-nosed dart, which is a fish. Right? Our adult worm lives in the intestine of this guy. This fish defecates. In the feces, it's the egg. The egg then is going to be consumed by our intermediate host. And our intermediate host is one of several species of snail. All right? In several species of snail. Now, once we get inside of the snail, that egg, let's do this, egg is going to hatch in the intestine, releasing the cotylocydium. Cotylocydium then is going to penetrate out of the intestine of that snail into the hepatopancreas, the digestive gland of the snail, where it will develop into a pre-adult. So we've got a pre-adult in the hepatopancreas. And it's going to sit there. It's going to sit there. Now this pre-adult, it's called a pre-adult instead of regular adult because it isn't quite fully developed yet. This life cycle stage has testes and ovaries. Ovary. it doesn't have eggs yet. So we've matured, sexually matured, but reproduction hasn't occurred to produce the eggs. So that's why we call it the pre-adult. It's this pre-adult stage that is our infective stage. Now what has to happen is that we have to get back to the snub-nosed darts. And that is where we have our host specificity. Because the snub-nosed dart has modifications in the pharyngeal plate that make them hard and allow them to crack and crush these snails and thus consume them. Not many fish have that. Not many fish specialize on snails, but the snub-nosed dart do. So, in this Lobatostoma, we are extremely host specific. It only infects these snub-nosed dart, which Trematoda, block-eye, it's a scientific name. That's the only host, the only fish host that is infected by these guys. That's it. You wipe out that host, you wipe out the parasite with it. So with our host specificity versus generalist and stuff, we've got host specificity. This is extremely host specific at the definitive host level, but it's more of a generalist at our intermediate host level. Questions? So, definitive host, snub-nosed dart. Got to be specific on this one. Snub-nosed dart. Intermediate host, snail. Is it required? Yep. Yep. It's an indirect life cycle, heteroxynous life cycle. We've got passive transmission, eggs to the snail. Passive transmission, snails to the snub-nosed dart. Although my guess is we haven't done a whole lot of research on this, so perhaps that pre-adult is altering behavior of the snail. Make them more exposed to predation. Who knows? All right, it's your first life cycle. You're gonna get more. My recommendation, kind of draw these things out a few times. All right, draw these things out. All right, we'll end it here. Don't forget quiz tonight, terminology. We will, I think that wraps up. Get this back up here real quick, in case you didn't get this. In case you needed the rest. We've got the definitive host, intermediate host, and we've got it. It's around the Great Barrier Reef. That's where we find them. Yep. You will. You can. Ultimately, yeah. So, the Wednesday before our exam, Wednesday, 8 o'clock, I think, is the deadline. If you didn't take it, it disappears, and all questions and answers become available at that point. Yep. All right.